Hello, everyone, and welcome back. We're going to go ahead and just go right into our last session, and hopefully everyone will be okay with that. We will hear from Dr. Kristin Herman, who is a medical geneticist with UC Davis Medical Center and the Mind Institute and Shriners Hospital for Children. So, Dr. Herman, I'll go ahead and turn it over to you. Thank you for being with us today.
Well, thank you very much for the invite. Today I'm going to talk about genetics, just to give you a very brief overview, because you can't understand genetic testing without having a basic understanding of genetics. So you can think of your genome, which is your genetic information, as a book. So your genome and all your genetic information, which codes all the instructions for all the mechanisms in your body, are contained on chromosomes. Next slide. And you can think about these chromosomes as chapters in that book. So a typical individual is going to have 36 chapters or 36 chromosomes. So you have 2 copies of each chromosome. 1 copy comes from mom and 1 copy comes from dad. And then females have 2 X chromosomes and males have 1 X and 1 Y chromosome. That makes females females and males males. And that becomes actually important in later on when you're talking about some genetic conditions. So on the chromosomes are hundreds to thousands of genes, and you can think of those genes as paragraphs in a book chapter. Next slide. And then within each gene, so each gene is going to encode specific instructions for a specific purpose, a specific protein, which is usually like a structural protein or an enzyme or things like that. So within each gene is a series of what's called introns and exons. And you can think of those as sentences within the paragraphs. So exons are actually containing the information for the protein sequence, versus introns contain other information, and they're actually cut out before the final protein coding instructions or mRNA is made. And if you look at the picture that I included a representation of a gene, you can see the exons are very small compared to the introns. And the exons, while having the most important amount of DNA as far as coding sequences are concerned, are actually a very small part of the whole genome, and represent a really small part of what our DNA is. However, again, about 95 to 97% of clinically relevant mutations or gene changes are going to be found in exons. So next slide. So when we go into genetic testing, there's different types of genetic tests. So first you can look at the karyotype, which a karyotype is going to look at the chromosomes under a microscope. And the purpose of this test is basically to make sure that all the chromosomes are present and there's no large pieces that are missing or duplicated. You can tell the different chromosomes by their banding patterns. So they're put under certain chemical environments, and you get this series of light and dark banding, and this series of light and dark banding can tell a cytogeneticist or an expert in the chromosomes which chromosome is which, and allows them to not only identify the chromosomes, make sure that they're all there, but also see, are there parts of that chromosome missing, duplicated, or switched around. So when you think of the book analogy, a karyotype is basically going to be looking at the book as a whole and going through it and looking to see if all the book chapters are there. And it can detect whether multiple pages are either absent or duplicated. Now, this is not going to be a common test that many individuals with who are dealing with an evaluation for a muscular dystrophy are going to see, but it is something you may come across prenatally when people are doing prenatal testing for things like Down syndrome, trisomy 18, trisomy 13, sometimes other conditions. And you can see the karyotype here. The book would have 47 chapters, as this individual has an extra copy of chromosome 21 and has Down syndrome. Next slide. So this is another test we actually don't use too much, but it can still be used, and it's something that I think you should know about. So FISH analysis, or fluorescent in situ hybridization, is a study that can detect missing or duplicated pieces of genetic material that are too small to see on the karyotype. So first of all, that's important. The most important information is you have to know what you're looking for, because each FISH probe is going to be specific for a region of chromosomes. And going back to the book analogy, a FISH is going to be able to detect missing or duplicated pages, and may even get down to, say, maybe some paragraphs. Again, we don't use FISH a lot, but if you're looking for something very specific, it can be helpful. So for example, with muscular dystrophy, if you have a large DMD gene deletion, FISH can be a quick and easy tool to see if that gene deletion is present or absent. Now on the screen, this picture may be a little bit small, a little hard to see, but when you do FISH, there are two probes that light up in two different fluorescent colors. So in this one, the kind of fluorescent yellowish probe basically says, yes, we are identifying the X chromosome, or everything worked as far as getting the probes to work in the lab properly. And then the red probe is the one specifically for the DMD gene. Or actually it's more of a DMD region, but anyway, so you can see that probe is present on one X chromosome, but absent on the other. So if the material is not there, the probe is not going to adhere, and you know that there's a deletion. Next slide. Now we're getting into studies that we use quite regularly, although again, maybe not so much in muscular dystrophy cases, but a lot of times muscular dystrophies are not identified right away if there's not a family history. And so a child may go through a regular hypotonia workup or something like that, or developmental delay evaluation, and chromosome studies such as a microarray are very common. So you may hear it said as a chromosomal microarray, you may hear a SNP array. But this study is going to detect deletions and duplications that are, again, too small to be seen on karyotype. And it uses a large number of probes along the chromosomes. And you can think of it as kind of FISH on steroids, that if you took, hundreds and thousands of FISH probes, then this is kind of what it is. So again, the chromosomal microarray is going to be, going back to the book analogy, they're going to be able to detect missing or duplicated chapters, pages, paragraphs, and actually you can even get down to sentences. So the chromosomal microarray now can detect gene deletions, and it can actually detect partial gene deletions. However, it's really important to remember that microarrays are not looking at the spelling sequences of the genes. They can see if the genes are there or duplicated. They can see if even exons or introns are there, but it is not going to look at the spelling sequence. So you can see on the photo, you can see the red is where saying that you do have 2 copies of that chromosome, and then the green area is where you only have 1 copy of that region of the chromosome. You can see how it goes to the picture of the chromosome below. And so this is a deletion of chromosome 20, the long arm involving bands 11.22 to 13.32. And actually, I kind of, this is the prettiest picture I could find on the internet, but I'm kind of cheating because actually this deletion would be large enough to be visible on karyotype. But again, we don't order a whole lot of karyotypes anymore, and a lot of these deletions are picked up by microarray. So here's, we're getting into the meat of what's going to be a lot of what you guys are going to be exposed as far as you're working up your children for various muscular dystrophies, the gene tests. So these tests are actually going to look at the spelling sequence of the genes. So if you can think of, again, back to the book, they're going to check the spelling of the words of the sentences. Depending on the lab or test ordered, this test can detect small and even sometimes larger deletions and duplications. The focus is usually on the sequencing of the exons, again, where the coding protein, or the coding information is. But again, depending on the lab and depending on the test ordered, sometimes you can get intronic variants or changes identified through these tests. So gene tests can include single gene testing, larger gene panels, or whole exome, or even whole genome sequencing, and I'll go through all those. So next. So single gene test is when you're looking at the spelling sequence or the sequence of one targeted gene. For example, if Duchenne muscular dystrophy is suspected, then a physician would order DMD gene testing. This test, single gene testing, is also helpful when you have a known variant in a family. So for example, say you have one brother who has DMD or Duchenne muscular dystrophy, and you have another child that you suspect Duchenne muscular dystrophy in. You know the genetic variation in the first brother, so you can test the second brother for that very specific gene change. You don't have to sequence the entire gene, but you can look at that known family gene variant, and it's actually much quicker and much cheaper to do as a general rule. So that's single gene testing. So then they have larger gene panels, and these gene panels are going to look at a larger number of genes. It could be anywhere from 2 to several hundred genes that are going to be associated with a particular clinical feature or diagnosis. So for example, congenital muscular dystrophy panel, congenital myopathy panel, seizure panel, intellectual disability panel, cardiomyopathy panel, skeletal dysplasia panel. So that all the genes are going to be related in some way to those overriding conditions or diagnoses. But it's important to remember that the number of genes and the specific genes included on each panel is lab specific. So if you have a muscular dystrophy panel at lab X, you may not have the same genes offered at lab Y. So it's important to really look through the genes and know which ones were on the panel before you do a comparison. So if somebody had, again, a gene panel at lab X, and they got certain results, say negative results, but another family member had testing it at lab Y, and they had positive results, you need to go back and make sure X had the gene that was found in lab Y to make sure that they are compatible, and that the person that had the negative gene testing was actually truly negative. Most labs are going to offer deletion duplication testing, and most will also look at least clinically significant intron variants in those genes. Labs are going to vary as to whether they offer complementary family testing for gene changes or variants of pathogenic gene changes or variants of uncertain significance. I'll kind of talk about what that means in just a minute. But it's really, as far as I'm concerned, it's very important to know whether the labs do offer this complementary family testing. As you know, that often when a gene change is found in a child, there are other family members that can be affected. So it's really nice when you can order that in selected family members free of charge. Also, again, if we have variants of uncertain significance, having family data can help us know whether that variant is clinically significant or not. I'll go through that in just a minute. There are some labs that will use duo or trio testing. So duo means that they have the proband or the person of interest, the targeted person being tested, and one other family member, or trio testing where they have 2 other family members that are included in the baseline testing. So you kind of get that complementary family testing up front. So there are a handful of labs that are offering that right away. Next. So here's the one that everybody hears about. This is kind of the big new fancy test that came out in about, clinically in around 2012, and that's whole exome sequencing. So whole exome sequencing is able to check the spelling or sequencing of pretty much all the genes at one time. And they're going to be focusing on exons because, again, that's where most of the mutations live. So the lab is going to use computer programs, usually for the initial pass, and then often, but not always, people, then to go through the data and try to figure out what out of the 20,000 plus or minus genes in our genome, what changes to those genes fit the reported clinical features. So there is error that can happen with whole exome sequencing. In that some people may, feel that a change is not significant when it ends up it is, or, vice versa. But a major limitation in whole exome sequencing is we just don't know what all the genes do yet. And variants in genes of unknown or uncertain significance are not going to be reported in a clinical whole exome test. So we currently understand what about, 35 to 40% of the genes do, as far as we know. This gene is associated with this clinical feature, things like that. So there's a lot of genes yet that we can sequence. We know the coding sequence of them, but we don't have a clinical feature or a clinical diagnosis associated with them yet. So that's probably the main reason why exome and genome sequencing can come back negative when we still highly suspect a genetic condition. So it's important to know that a negative exome does not rule out a genetic condition, and there are these limitations. A lot of labs now, a lot of labs now can detect deletions and duplications that would be found on a microarray. So there's, this is a little bit more of a one-stop shop in some ways. Some exome sequencing now can look at limited introns, but again, exons are the primary focus of whole exome sequencing. It is vastly preferred that trio testing be done in whole exome sequencing. And usually we ask for the parents if the parents are available. Sometimes parents are not available and we can use one parent and a sibling or something like that. But this is really important. Having the parental data up front is super important with whole exome sequencing, more than the gene testing, to help clarify and classify the results. And so when you're wading through a lot of data that you get for whole exome sequencing, knowing if a variant is inherited, if it's inherited as we would expect it, or if it's a new mutation, helps know whether a particular gene change is really clinically significant or not. Now, the rate of finding a diagnosis currently in whole exome sequencing is about 30 to 35%. And you'll notice that's about the same as the genes we understand. So again, there's a lot of people that will have whole exome sequencing that we suspect a genetic condition, and it comes back negative. And that's either because the whole exome sequencing is not designed to detect the type of mutation that a patient has, or we just don't know what that gene is, and the genetic cause of an individual is a gene that we don't understand. So next slide. So similar to whole exome sequencing is whole genome sequencing or whole genomic sequencing. Very similar to whole exome sequencing, but there are some added benefits. One is it actually will sequence all the introns, and so we can look at intronic mutations. There are also, theoretically, it's supposed to be able to be done in all labs, but there are certain labs that are still trying to work at getting validated for it. But it's supposed to look at trinucleotide repeat expansions, and that's kind of a whole nother topic. But essentially, these are genetic changes where the normal gene will have a certain number of repeated words, if you think about it. So repeated words. And if there's too many repeated words, then you have a disorder or a genetic condition. So whole exome sequencing cannot look at trinucleotide repeats. Whole genome sequencing is able to look at trinucleotide repeats, although not all labs are able to do that right now. And whole genome sequencing can better sequence some of the areas of the genome that are difficult to sequence when you're using the whole exome platform. So it does a little bit better at detecting mutations. Again, the similar limitations are mostly that we have a lack of knowledge of a large number of genes. So you'll see with whole genome sequencing versus whole exome sequencing, the diagnostic rate is about 5 to 10% more, because it is able to detect some of these other mutations. Oh, and I forgot to say, with whole genome sequencing, pretty much regardless of the lab, it can detect the duplications and deletions that you would otherwise see in a chromosomal microarray. So really, it is hoped in the future that whole genome sequencing would be a one-stop shop kind of test for kids that we really don't have a great idea of what's going on. Next slide. So there's a couple things if you have whole exome or whole genome sequencing that you have to know about and should be counseled about. So there are certain things called secondary findings. And secondary findings is a list of mostly gene predisposition disorders, such as cancer predisposition syndromes, cardiovascular predisposition syndromes, and maybe a couple others. But those are things that you have an increased risk of developing a certain condition. So for example, like the BRCA1, BRCA2, which are breast ovarian cancer genes, those are cancer predisposition syndromes. It doesn't mean that if you have a mutation, you're absolutely going to get cancer, but it does greatly, greatly increase your risk over the general population. So it's been determined by the governing bodies of medical genetics that there is this list of secondary clinical diagnoses, and you can, and if whole exome or whole genome testing is done, even if it has nothing to do with the reason for the test, the lab will report disease or likely disease causing changes in any of these secondary finding genes if the patient opts to have them. So you should be counseled if you have whole genome or whole exome that you can opt in or opt out of learning about these secondary findings. Now, it's also important to know that variants of uncertain significance are not going to be reported in these genes, only things that we know cause, or we're pretty darn sure cause disease. If the whole exome has duo or trio testing, the family samples will then be looked at to see if the variant's inherited. So first they're going to see if it's present in, say, the child or the proband. And if they find, say, a BRCA1 variant, pathogenic variant in the proband, then they'll look at mom and dad's sample and see if it's inherited. So they don't necessarily, and this can be a little bit lab dependent, they don't necessarily look at the parents for these secondary findings if they don't find anything in the child. Now, there are a couple labs that will report out secondary findings in the parents, even if the child didn't end up inheriting it. But it's really important to know that there are some labs that if they don't find any in the child, they're not going to look at the parent samples. So some people will think, well, I had a negative parental exome report, so I'm free of any of these secondary findings, and that may not be true. There are some labs, one in particular I can think of, that also will offer carrier testing if the patients and the parents want it, which would be looking for carrier status, meaning you have one mutation of an autosomal recessive disease. An autosomal recessive disease is a genetic condition where you need a mutation or a gene change on both copies of a particular gene in order to have the disease. If you have only one copy, you're totally normal clinically, but you are a carrier. And knowing if you're a carrier or not can know if you're at risk for having an affected child in the future. So some labs will offer the carrier testing if the parents and child opt in, and also pharmacogenetic testing. And this is usually on whole genome testing that is offered. And pharmacogenetic testing is looking for particular variants that may have play a factor in how you metabolize certain medications. And again, that's kind of a whole nother lecture, but it's worth mentioning. Next slide. So kind of a big question is, what test is appropriate to be ordered? And I would say that it depends on what you're looking for. And also, unfortunately, insurance is often a major factor. It shouldn't be in an ideal world, but it is. So everybody likes the latest and greatest. And so whole exome and whole genome seems like, you know, why don't we just order those? Well, bigger is not always better. If you have a fly, you don't want to use a cannon to try to get rid of it. So if you know a family variant, or you know you're 99.9% sure you know what condition you're looking at, single gene testing is important. If you have a known familial variant, there's no need to do whole exome. You test for that particular variant, then the test can conclusively diagnose or conclusively rule out the condition. There's no need for larger testing. Again, if a clinical feature points to a specific gene, a specific condition, then you order that single gene testing. So if you know a patient has Duchenne muscular dystrophy, but there's no family history to know which specific gene change is responsible, or there's a family history and nobody's ever gotten genetic testing before, but you know it's Duchenne muscular dystrophy, then look at the DMD gene. You don't need to do whole exome sequencing. If a clinical feature points to a specific group of disorders, but there's multiple genes, then a gene panel may be more helpful. So say you think it's a muscular dystrophy, but you're not 100% sure what type, or say like limb-girdle muscular dystrophy, there's a large number of genes that can cause a very similar phenotype. Well, then doing a panel may be better. Often because these panels are targeted to a specific disorder and they are working with a smaller number of genes, it's just easier to work with that smaller amount of data. They have better coverage of those genes in order to detect any mutations that are there. And so sometimes gene panels are very helpful, and quite honestly, a lot of times I can get insurance to cover a gene panel, but they don't often like covering whole exome sequencing. Gene panels are much cheaper, and a lot of times I can get insurance to cover it when they would not cover other things. So if your clinical features are not clear, they're not really fitting into a particular disorder, or you're thinking maybe a person has more than one condition, then absolutely 100% whole genome, whole exome sequencing is appropriate and called for. And there are a lot of cases that this is the preferred method of testing. So again, in cases of suspected muscular dystrophy or myopathies, chromosome studies are not often indicated, but if the muscle findings could be part of a potentially larger chromosome disorder, then it may be worth doing chromosomal microarray studies. However, if the insurance will cover it or the patient has resources, whole genome sequencing may also be a better option, because you'll get that chromosome study in addition to sequencing multiple genes. And again, there are some cases that will think, this is not completely fitting into a known condition, and are we looking at multiple genes being affected? And sometimes it's not a chromosome problem that's causing multiple genes to be affected. It's just actually multiple genes are affected. Next. So with any genetic test, there are three possible results. You can get a positive test, or what we call a diagnostic test, and that's when a pathogenic or likely pathogenic variant is identified. That means that the change to the DNA does cause disease. This will make or confirm a clinical diagnosis. Then you can get a negative test, and that's when they see no genetic changes or no changes to the DNA, or changes to the DNA that we know are benign, that we know very well do not cause disease, and those benign changes are not going to be reported. It's important to remember a negative test result does not always rule out that particular condition, nor does it mean that that condition is not genetic. It just means we looked at the available genes and we could not, with our current information, we could not find an answer. Then there's the variant of uncertain significance, or an uncertain result. This is a change to the DNA, and we just don't have enough information to know whether the variant is disease-causing or not. It's important, if you find a variant of uncertain significance, it is not diagnostic. You should not act upon a variant of uncertain significance until there's enough evidence to be able to classify that variant one way or another. Family studies can be really important. Functional studies can be helpful, and also time. Figuring out more information about the variant through the labs, through other people testing, through population data, things like that can often help classify variants of uncertain significance. Next. So I just wanted to run through really quick some examples of some certain variants, and it's kind of hard to translate DNA words and letters and sentences into English, but this is kind of a sort of an example. So if your baseline sequence is the cat was eating chicken, and you had a DNA change that made it the elephant was eating chicken. Well, first of all, the elephant is a whole lot bigger than a cat, and is probably because the size difference is going to make a difference in the resulting story or the resulting protein. Also, while it makes sense that a cat would be eating chicken, it doesn't make sense that an elephant would eat chicken. And so you can see that there's certain things in that DNA change that is going to suggest, yes, this is disease causing. This is going to mess up the resulting protein. Another is, okay, that you have the cat was eating chicken, and then you have the eating chicken. So you can see cat is deleted. And now all of a sudden you're what's eating chicken, and it doesn't make any sense. Then you have the cat was eating chicken and the at was eating chicken. So the C was deleted. And again, the at was eating chicken, that doesn't make sense. So you can see how there's two different deletions, a very large one and a very small one, but they still can result in things not making sense. And then you have the cat was eating chicken and the cat was eating chicken, please cut this out. So what happened in this sentence is the please cut this out was supposed to not be in the final part of the protein. It was an intron, but the instruction, there was a mutation in the instruction site that said, get rid of this intron before the mRNA was made, and that did not happen. So now all of a sudden you have a lot of extra DNA affecting the protein. An example of a benign variant would be the cat was eating chicken or the feline is eating chicken. Cat and feline are basically the exact same thing, so there's really no difference in the final instructions. And some examples of a variant of uncertain significance. The cat was eating chicken or the dog was eating chicken. Well, not really sure about this one. You know, cat and a dog could be about the same size. It makes sense that a cat and a dog would eat chicken. So is the difference of a dog enough to cause a disease or not? And you can see that that's how things would be questioned. And then you can see the cat was eating chicken versus the cat was eating fish. Again, it kind of makes sense. A cat would eat chicken, a cat would eat fish, but is that difference between fish and chicken going to be enough to cause a genetic condition or not? So those are some kind of examples of genetic test results and spelling and sequencing errors that can cause disease, cause benign variants, or variants of uncertain significance. And I believe that's my last slide.
Yes, it is. Excuse me. Thank you, Dr. Herman. We did have a couple questions. We had some chatted in and one in our Q&A box. If I had a genetic test 10 years ago and it was inconclusive, do you suggest that I get another one done?
Yes, absolutely. Things change a lot in 10 years. The methods of how the genetic testing is done, you know, how variants are called. So say something was a variant of uncertain significance 10 years ago, we may have enough evidence now to reclassify it. Yeah, absolutely. I would, well, let me put a little bit of a caveat on that. If it was say it was a variant of, so if it's inclusive because there was a variant of uncertain significance, then they may just be able to, there's certain databases that physicians or especially geneticists can go to see if that variant has been reclassified or not. But if it was inconclusive just because, you know, just didn't really find anything, then absolutely redo it because, again, new genes, new technologies, yeah.
Okay. And are these blood tests or saliva tests or both?
They can be, it's on DNA. And the nice thing is DNA is everywhere.
Okay.
DNA is in blood, DNA is in saliva, DNA, you can do a buccal sample. So yeah, it doesn't matter as long as it's DNA. The nice thing is, even though say you have certain instructions that are looking at your eyes, those same genes are in your saliva cell or your cheek cells, those same genes are in your blood. So the DNA is the DNA, it just depends on which organ system, if they're turned on or turned off. But the same gene sequencing is there. So no, it doesn't matter what the sample is, and different labs are going to be validated for certain samples. I personally, because I do a lot of the sample collection myself, especially at Shriners, I don't even work with labs that take blood samples only. I will only work with the ones that take saliva or buccal because it's so much easier. You can send kits to the patient's house, they can collect it themselves. Okay. It's much easier. But yeah, there's no difference with the sample.
Okay. We have a question about insurance, and they're asking, are insurance companies more open to coverage based on a family history criteria?
I wish that was the case.
Okay.
It's just hit and miss with the insurance.
Okay. All right.
If there is a family history and you're doing known familial testing, again, that's cheaper. So usually I can get the insurance to cover it because it's a much cheaper test. So there's so many variables. It's so annoying. It takes so much of my time as a geneticist to figure out, what insurance covers what test, and if it's denied, then trying to do appeals, and it's very frustrating, but that's just the landscape we live in right now.
True.
But yeah, sometimes, family history is enough. Some, they just don't want to pay for it, period. And it takes a lot of work or an act of God to get them to cover it.
Okay. All right. Well, I don't see any more questions in our chat or in our Q&A. So thank you very much, Dr. Herman. We appreciate your time this morning.
You are very welcome. And I was very honored to be asked to come and speak with you all. And thank you again for the invite.
Thank you.
All right.
And this concludes our seminar for today. I want to thank all of our presenters for their time and effort they spent putting together their presentations for this Saturday. I would also like to thank our seminar supporters, Biogen, Numotion Foundation, and Genentech. And thank you to you guys for taking time to join us today. And I hope you found it helpful and informative. If you have any questions after the seminar, please email us at mdaengage@mdausa.org. And finally, I wanted to remind everyone I will be sending out a survey from this email address that will have some information on it as well from our supporters. And so we appreciate you filling out that survey and giving us your feedback. And thank you again for joining us. And we hope you enjoy further seminars from the Engage program. So thank you and enjoy the rest of your day. Bye-bye.